Welcome to the Chemistry, Biology and Math Revision Hub. Today we're doing the Pearson Excel International A-Level Biology Unit 6 for January 2024. Let's begin with the first question. In question 1, they said the photograph shows parts of Tussilago farfara. This is a native species of plant that grows in North Africa and Central Asia. So this is the plant they're talking about. They say this plant can be used as a medicine. They want you to describe how the antibacterial activity of different parts of this plant could be compared. In this case, because they've said different parts of the plant, you could choose to use roots, stems, leaves, and so on. My description here, I had to find ways of extracting the antimicrobial agents from the specific parts of the plant that I've chosen. Then you can grind in order to extract the reagents or the chemicals found inside, and then culture some bacteria on agar plates and compare the diameter of the zone of inhibition after putting the antimicrobial agents on the petri dishes that were pre-cultured with bacteria. So I said, use at least 2 different parts of the plant. It could be roots, leaves, stems, or flowers. Then you extract the antimicrobial agents by grinding the chosen plant parts in a suitable solvent. Use the same volume of solvent for all extractions, and then use aseptic techniques during the culture and inoculate bacteria on agar plates. Then soak filter paper discs of similar size into the same volume of the extract for the same time. Use sterile forceps and transfer the soaked filter paper into the inoculated agar plate. And of course, you have to make sure this filter paper is not dripping, otherwise the concentration transferred could be different. Culture for at least 24 hours at about 26 degrees Celsius. The temperature has to be at least between 25 to 30 degrees. And then measure the diameter of the zone of inhibition around the filter paper for comparison. Then you repeat each experiment and calculate the mean diameter. In case you use the liquid culture, you will inoculate with the same volume of bacteria and then add the same volume of the extract. You culture for 24 hours at 26 degrees Celsius and measure the turbidity for each extract. You repeat cultures to get the mean and compare the results. The one with least turbidity will have the best antimicrobial activity because it would have grown less bacteria. In part B, scientists have isolated antibacterial compounds from plants such as Tussilago farfara. State what is meant by the term bacteriostatic and bactericidal antibiotics. Bacteriostatic antibiotics prevent increase in the number of bacteria, and bactericidal antibiotics of course kill the bacteria. And next they explain how the code of practice for prescribing antibiotics helps to control infections. With this code of practice, the doctors are required to prescribe antibiotics only when required, and the patients are required to complete every dosage. Otherwise, there is a chance of antibiotic resistance. So I said antibiotics can only be prescribed when necessary for bacterial infections, and this creates less selection pressure for bacteria, lowering the chances of mutation to form resistant bacteria. So this brings us to the end of question 1. Let's continue to question 2. In question 2, the photograph shows a pollen tube growing in a culture media. So this is the pollen tube. And down here they say pollen grains were placed on glass slides covered in growth medium. 8 slides were incubated at 24 degrees Celsius. After 1 hour, 1 slide was placed under a microscope and the length of 10 pollen tubes were measured. The measurements were repeated at hourly intervals with the remaining 7 slides. The graph shows the results. On the vertical axis, we see the mean pollen tube length and time in hours on the horizontal axis. And we see as time increases, the mean pollen tube length is also increasing. So the first question says, calculate the mean rate of growth of the pollen tube between 1 and 5 hours. They want you to include units in your answer. So you have to get the values at 1 hour and the values at 5 hours. At 1 hour, we have 140, and at 5 hours, we have 120. So the mean rate is going to be 120 minus 40 divided by 5 minus 1, which is 80 over 4, and the answer is 20 micrometer per hour. That is what we have here. In part B, explain how changes in temperature could affect the rate of growth of pollen tubes. Of course, the higher the temperature, the higher the kinetic energy, more collisions between the enzyme and the substrate, and then a higher rate of reaction that could lead to growth to occur faster. But if the temperature is beyond the optimum, then the rate is going to slow down because enzymes are going to be denatured. So I say the higher the temperature, the higher the kinetic energy, the higher the rate of formation of enzyme substrate complexes. And of course, this is going to increase the rate. So we will have faster cellular or biochemical reactions like respiration, and then more cells will divide and the pollen tube will increase in size. However, after the optimum temperature, increase in temperature denatures the enzymes, leading to slowed growth. In part C, abiotic variables were controlled in this investigation. State 2 abiotic variables other than temperature that could affect this investigation. The first variable I chose is pH, and the second one could be mineral concentration in the growth medium. Then they say choose one of the variables you have identified in 1 and describe how this variable can be controlled. The variable I chose was pH, and we can control that using a buffer. Next, they say suggest what effect not controlling this variable would have on the results. If pH is not controlled, it's going to change, and pH change would affect the hydrogen bonds in the enzyme. And this would change the 3D structure of the enzyme, meaning it's going to be denatured, and this is going to lead to a slower or decreased rate of reaction. In part D, describe how this method can be modified to investigate the effect of age of pollen on the rate of growth of pollen tubes. This could be carried out by storing pollen for different periods of time and then germinating it to see how the growth rate is going to be. So I said pollen can be stored for different periods of time and periodically germinated. And during storage, the conditions should be the same and the pollen should come from the same source. So this brings us to the end of question 2. Let's continue to question 3. Question 3, the photograph shows a lizard native to a Caribbean island near the equator. This is the lizard. They say this small lizard lives in tropical vegetation between 1 meter to 3 meters above the ground. This lizard uses specialized toe pads to hold onto the vegetation. A scientist sampled the lizards along a 2-kilometer transect. The toe pad area of the front limb of each lizard was measured. Remember, only the front limb was measured. The lizard was then released. A hurricane struck the island with the highest wind ever recorded. The scientists repeated the sampling method along the same transect after the hurricane. The results of this investigation are shown below. So we can see the surface area in millimeters squared of the toe pads before the hurricane, and then we can see that after the hurricane in sample B. And they say state a suitable null hypothesis for this investigation. Since we are comparing surface area into regions, so I say there is no significant difference between the surface area of the toe pads before and after the hurricane. In part B, they say draw a suitable table to display the data and your calculated means for the toe pad area of sample A and the toe pad area of sample B. This is how presented my sample. We have sample A, the values are there, and the value for sample B. My means for sample A, I have that, and here I have that. So the lowest value here is 0.7, the highest value is 1.6, the lowest value here is 0.8, the highest value is 1.9, so we can compare the lowest and the highest with the mean values that were obtained. In part C, draw a suitable graph to show the mean toe pad area for sample A and for sample B. Include an indication of the variability of the data. When they say variability of the data, we have to include the error bars. So here I drew the graph. This is my mean value for sample A, and that is my mean value for sample B. But then I included the lower value or the least value for that, and the highest for that, the least value for that, and the highest for that. In this presentation of my data, I've included the error bars, and my axes are labeled. I have units, and I have chosen suitable scales. So this is how you could present your data to get the 3 marks that are awarded here. In part D, the scientists analyze the data with a t-test using the formula. So this is the formula used. N is the number of samples for each treatment, and they've given us these values. They want us to calculate the value for T. So I chose this to be the X for sample A, and that is going to be the mean for sample 2. So 1.2 minus 1.5 divided by the square root of that, divided by 9, which is the total, plus that divided by 9. I got negative 1.96, but of course I can ignore the negative, because if somebody said 1.5 minus 1.2, then they'll get a positive value. So the positive value for T is 1.96 to 3 significant figures, and that's what I wrote here. The next part says the table shows the critical values of T for different degrees of freedom. The number of degrees of freedom are calculated by saying Na minus 1 plus Nb minus 1. Since each number is going to be 9, so 9 minus 1 plus 9 minus 1 gives us 16. So when I look at my table of critical results, at 16 degree of freedom, at p is equal to 0.05, my value is going to be 2.12, and that is the critical value, which I'm going to compare with a calculated value. And next they say describe the conclusions that can be drawn from this investigation. They want you to use the information in the table to support your answer. If I compare the calculated and the critical value, the calculated value is 1.96, and this is lower than the critical value, which is 2.12. So I will accept the null hypothesis, and this means there is no significant difference between the surface area of the toe pads before and after the hurricane. In part E, describe how the scientists could extend this investigation to collect more data to either support or reject the hypothesis. Here, what the scientists could do is to repeat the studies in other areas. They could also use other species of lizards in order to compare. And again, because in this experiment they measured the forelimb, they could also measure the hind limbs and then compare if there was a difference before the hurricane and after the hurricane. So this brings us to the end of question 3. Let's continue to question 4. Question 4. The photograph shows half a watermelon fruit containing seeds. We can see the seeds in the watermelon. They say watermelon plants are grown in all parts of the world. Watermelon seeds can be diploid with 22 chromosomes or tetraploid with 44 chromosomes. Tetraploid seeds have different food stores and thicker seed coats than diploid seeds. Watermelon seeds respire their food stores during germination to develop into seedlings. A student formed the following hypothesis. Tetraploid seeds have a higher respiratory quotient than diploid seeds. So they want you to plan an investigation to find evidence to support or reject this hypothesis. In part A, they want us to describe preliminary practical work that you might undertake to ensure your proposed method would provide quantitative results. In the preliminary practical work, you usually find out which conditions are going to be suitable for varying the independent variable, or you're going to measure the dependent variable. In this case, maybe for the respiratory quotient, we want to find the volume of CO2 that has been released and then the volume of oxygen that has been used up. So you have to find ways in which you're going to measure the volume. And of course, we have to find ways of which seeds are we going to use for, how many seeds are we going to use in each experiment, and so on. So I said, choose suitable seeds to be germinated, find a suitable mass or number of seeds to be used, also find suitable germinating conditions like temperature and pH, find a suitable method to measure the volume of oxygen used and the volume of carbon dioxide produced, and then find a suitable method for absorbing carbon dioxide produced. In part B, devise a detailed method including how you would control and monitor important variables. You begin by stating the dependent variable. Here, the dependent variable is the volume of oxygen used per time. Set up the respirometer with an attached syringe. This syringe could be used to refill the gas in order to find out the volume of oxygen that has been used up, and a capillary tube containing a colored solution. You can use seeds of the same mass or age. Use soda lime to absorb the carbon dioxide produced so that the volume of oxygen used can be accurately measured. Allow the seeds to respire for a specific period of time, meaning in each experiment that time has to be kept constant. Use the syringe to fill the volume of oxygen to return the colored solution to its original position in the capillary tube. The volume used to refill or the volume used to return the colored solution to its original position is the volume of oxygen that has been used. So allow the seeds time to acclimatize. This is to allow them to get used to the new conditions before we begin to measure, meaning in each fresh experiment. Repeat the experiment without soda lime for the same period of time and record the volume of air required to return the colored solution to its original position. The volume of carbon dioxide produced is equal to the volume of air required to return the colored solution to its original position with soda lime minus the volume required without soda lime. Then we can calculate the RQ, which is the respiratory quotient, by using the volume of carbon dioxide produced divided by the volume of oxygen used. Repeat the experiment with other seeds under similar conditions. Keep the temperature constant using an air conditioner and ensure that you use a safe antimicrobial to disinfect the seeds before use. Here I described another method somebody could use to get a volume. The capillary tube is going to be in shape of a cylinder and volume of a cylinder is pi r squared h. this distance moved by the colored solution is going to be proportional to volume. We can use the distance moved by the colored solution to represent volume. Volume of oxygen used is proportional to the distance moved by the colored solution in the presence of soda lime or potassium hydroxide, whichever you choose. So the volume of carbon dioxide produced will equal to the distance moved by the colored solution in the presence of soda lime minus the distance moved by the colored solution in the absence of soda lime. In the end, respiratory quotient can equal to volume of carbon dioxide produced divided by the volume of oxygen used. They say describe how your results should be recorded, presented, and analyzed in order to draw conclusions from your investigation. So here you don't need to draw anything, you can just describe what you will do. So I said present the results in a table with headings and units, include a column for the mean, then draw a bar graph with labels and units. Then use an appropriate statistical test like the t-test to test the significance of the results or to establish significance of the results. In part D, suggest 2 limitations of your proposed method. Of course, it's going to be harder to measure volumes of gas that were used, or like in this case, volume of carbon dioxide produced and volume of oxygen used. It's going to be difficult to control or prevent contamination of the seeds, and returning the colored solution to the original position could cause errors. This is when you're determining the volume of gas that has been used or produced, so there could be some errors in the estimation. So this brings us to the end of question 4, as well as to the end of the whole paper. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.